My name is Pathak, and today my topic for protein expression presentation is vector. So the content that we're going to deal with is the background, the problem statement, cloning strategy, protein expression, protein purification, activity assays, and the feature class. So coming to the background, what is melittin? It is a principal toxic component in the venom of the European honeybee Apis mellifera, and it is a cationic hemolytic peptide. So the peptide consists of 26 amino acids with the sequence, this one. It has several important biological effects and has a relatively low toxicity. It has no disulfide bonds in which the N part of the terminal is hydrophobic, while the C terminal is the hydrophilic, and it is strongly basic. So what is the problem? So antimicrobial resistance happens when germs like bacteria and fungi develop the ability to defeat the drugs designed to kill them. The resistant infections can be difficult and sometimes impossible to treat. With the emergence of antibiotic resistance, researchers have recently focused on antimicrobial peptides as potential drug candidates. So the cloning strategy that we're going to use is using PHAR as the expression system for cost reduction. E. coli can also be used, but I did not use it as it has several disadvantages, including bacteria's LPS contamination, as well as product toxicity for the recombinant E. coli. The PPIC9 vector was used to transfer melittin promoter to PHAR. This is the vector that we're going to use. It has three major components: the alpha factor secretion signal, the HISPO gene, and the alcohol oxidase promoter. This one. The presence of them enabled me to use a minimal medium for expression and to extract the secreted form of melittin from yeast cells. As P. pastoris naturally lacks significant secreted protein, the total protein amount of the supernatant is associated with the recombinant peptide when a culture medium is lacking protein and peptide is used. This food enables P. pastoris to grow in a peptide-free culture medium. The fragment that we're going to use is this one, the melittin from Apis mellifera mRNA. So it for the optimization of the fragment, that is the gene of interest, nantrine was used. The optimal sequence entered the PPCIN vector using the NOT1 and the ZO1 restriction enzyme. Some other sequences were also added, the ZOL and from the ZOL and the NOT1 restriction sites. The alpha factor sequence that would be removed after digestion of ZO1 and two stop codons. So this is the sequence. This is the promoter sequences, and this is the forward and the reverse one. You can see the annealing temperature and the melting temperature as well. This is the vector and the melittin are ligated into it on these sides, on the multiple cornea side, and this was prepared using SNAP gene, as I told you. So as I told you before, this is the map that was used. This is the vector and this is the inserted fragment. This is the fragment at the ZO1 and NOT1 sites. So PJ's digestion product was actually electrophoresed and melittin was extracted from the agarose gel 2%. To ligate the gene cassette into the PPIC9 vector, a mixture was prepared containing 90 nanograms of the digested PPIC9, 30 nanograms of the fragment extracted from the gel, the agarose 1, 1 unit of the T4 DNA ligase enzyme, and 1 microliter of 10x ligation buffer. The ligation mixture was incubated for 16 hours at 14 degrees Celsius. So the PCR product was then ligated into the vector. To confirm the ligation accuracy, a PCR reaction was performed with AOX primers and finally sequencing was done. The PCR reaction involved the denaturing, annealing, and extension. These were done at different temperatures. So as I told you, the verification of sequence was done by the agarose gel to see if the PCR product was of the correct size. This is the apparatus. So before the transformation of the cell, the preparation of PPIC9 vector to be ligated into the P-pasteur's genome is to be done. Vector linearization is very important, and enzymes like Saxial facilitates this, the integration of the recombinant vector into the P-pasteur's. Therefore, here we use SAC1 with 1 ml of enzyme per 1 microgram of DNA ratio, which was used for 1 hour at 37 degrees Celsius. The vector was then purified using silica bead DNA gel extraction kit. The concentration and purifier of the obtained DNA were measured by the thermoscientific nanodrop 100,000 spectrophotometer. Then the transformation of the cells happen using the gene pulse apparatus and for electroporation. The PHAR from minus 70 degrees Celsius stock was used to prepare the competent yeast cells, inoculated at 220 ml of YPD medium and incubated at 30 degrees Celsius and 200 RPM, shaking for 272 hours. Then the competent cells were mixed with the linearized, in the previous slide, the linearized PAPIs in melittin vector, which contained 10 microgram of DNA, and moved to a 0.2 centimeter cuvette for the electroporation, which was done using the gene pulse apparatus and incubated at 4 degrees Celsius for 1 hour. The yeast cells were dissociated from the medium and the pellet was washed again by cold distilled water and sorbitol 1 m and centrifuged at 4000 rpm for 5 minutes. Finally, the pellet was dissolved in sorbitol 1 m, 300 microliter, and each colony was collected and suspended in distilled water to confirm the transformation. So for the protein overexpression, these cells can be transformed on a histidine-free medium as well, as they contain histogene already. So the 100 ml of the suspension was distributed on a minimal dextrose medium using these following chemicals and incubated for 3 to 5 days at 30 degrees Celsius. For the removal of the remaining extracellular DNA, washing was performed 3 times by distilled water and centrifugation was done at 8000 rpm for 5 minutes. The final suspension has the OD 2 to 3, which was measured on the CD apparatus, and 1 micro ml of was used as a template in colony PCR by AX the primers. For the screening for selection of highest melittin production, the concentration of methanol, pH, and time effects were evaluated on S. aureus, as you can see on these slides, and the selected colony regarding the melittin expression was seen on these. To investigate the methanol concentration effect, the BMM medium, which contained a different percentage of methanol, was used to compare the inhibition zone after 48 hours of expression. To evaluate the pH effect, the inhibition zone was compared in a medium without the buffer and also with the buffer with different pHs. The time effect was finally compared at 24, 48, and 72 hours under optimal values of methanol concentration and pH. After the selection of screening of the best transformants, the protein purification was done. So for the protein purification, after the biomass production in the BMG medium and investigating the optimal conditions of methanol concentration, pH, and time, like the previous slide I mentioned, centrifugation at 1000 rpm for 15 minutes was done using the containing pelletin from the yeast cells. This was done using the Vivaspin 20O apparatus. These separated the large protein molecules which are larger than the 5 kDa, and then there is complete removal of the remaining proteins such as that released from the disrupted yeast cells in expression. By this method, the melittin remained in the solution, and as its molecular weight is less than 5 kilodalton. To determine the solution concentration, Bradford assay was performed, and the solution was then lyophilized for storage. These lyophilized storage and can then be used for the activity assay to evaluate the antimicrobial activity of the recombinant melittin, values of minimum inhibitory concentration and also the minimal bactericidal concentration were investigated on 5 gram positive and 12 gram negative bacteria. Broth microdilution method using the lyophilized powder of melittin was performed for this purpose. This is the broth microdilution apparatus. And then the production of melittin by this method costs very less, and it paves the way for further studies and also for the therapeutic usages of the melittin. So the growing problem of drug resistance, it poses as a major public health challenge, and so accumulated evidence shows that protozoan parasites such as Plasmodium and Toxoplasm susceptible to melittin at micromolar concentration. So what the melittin does is it obliterates these pathogens by several mechanisms of actions, like the disruption of cell membrane, the destabilization of the calcium homeostasis, and the reduction in mitochondrial membrane potential. So in this way, the melittin actually helps in fighting off the antimicrobial resistance. So, apart from the antimicrobial resistance, melatonin can actually be used in different things. A conjugation of melatonin with nanoparticles was great, promises a great in different biomedical applications. Not only this approach improves the target specific delivery of melatonin with less cytotoxicity, but it also enhances the proteolysis stability of nanoparticle melatonin conjugates. Several other anti-cancer as well as anti-diabetic properties of melatonin are currently being studied, and thus melatonin can be used in different places for as a drug. If you have any questions, feel free to email me. And yeah, thank you so much, guys. Have a nice day.